Chapter 5, Membrane Dynamics. In this chapter, we'll talk about mass balance and homeostasis. We'll look at diffusion and then other modes of transport like protein-mediated, vesicular, transepithelial. We'll also discuss osmosis and tonicity, and then we'll end with the resting membrane potential and use insulin secretion as an example of how all of these things work in our body. So the law of mass balance says that if the amount of a substance is to remain constant, any gain must be offset by an equal loss. So quite simply, if you eat more than you lose, so you bring in more calories than you burn, you're going to gain weight. If you want to stay the same size, you want to eat calories and burn calories at the same rate. Homeostasis, a concept that I said is going to come up throughout this term, when our body is working happily, when everything's the way that it's supposed to be, it does not necessarily mean everything is equal. So some things are. For example, we have osmotic equilibrium. So osmosis has to do with water. This is the total amount of solutes per volume of water, and that will be equal across a cell membrane. On the other hand, we have a chemical disequilibrium and an electrical disequilibrium. So the example of a chemical disequilibrium would be sodium and potassium. Sodium is much higher extracellularly compared to intracellularly, whereas potassium is much higher inside the cell compared to the outside. They are not equal across the cell membrane, but that doesn't mean we are not in homeostasis. We have this chemical disequilibrium, but we are still in homeostasis. Also electrically, we have a disequilibrium. The inside of a cell is more negative than the outside of a cell, but we are still in homeostasis. Now let's talk about diffusion. And this is a nice figure that goes over different types of movement, and it shows you the energy requirement, meaning do we need ATP, do we not? And then it looks at the physical requirement. So the simplest form of diffusion is called simple diffusion. It's the simplest one. This is where a solute moves across a cell membrane from an area of high to low concentration until equilibrium is reached. And it simply goes through the lipid bilayer. So is there a physical requirement? No, there isn't. The molecule will just go through the lipid bilayer. Is there an energy requirement? There is not. No energy, ATP is required. You just diffuse from high to low. Facilitated diffusion is similar to simple diffusion, meaning you're going to move a solute from high to low until you reach equilibrium, but you need a facilitator. You need a helper. You need some sort of a protein in the membrane. So there is a physical requirement, a protein, but you don't need energy. Again, diffusion, high to low, that's what drives the movement. Then we have forms of movement where energy is required. The first being primary active transport. So the example of primary active transport will be our sodium-potassium pump. To pump sodium and potassium, ATP is required. So that's an example of primary active transport. We also have secondary active transport. Secondary active transport requires energy, but it requires energy indirectly. So notice there's no direct line from ATP to secondary. Instead, secondary active transport that we're going to learn about, called the SGLT, sodium glucose link transporter, depends on our sodium-potassium pump, which is an example of primary active transport, to create the concentration gradient necessary for movement to occur. So that's why it's called secondary active transport, because it doesn't use ATP directly. It needs it kind of indirectly. All of these have a physical requirement. There is a protein in the membrane that helps this occur. Then we have endocytosis, exocytosis, phagocytosis. ATP is required for all of these types of movements, and we use vesicles. So endocytosis is when we're going to bring something into the cell. Exocytosis is where we're going to release something from the cell out into the extracellular fluid. And then phagocytosis, think of your immune cells phagocytosing or eating. Now diffusion, there are some properties I want you to know. One is it's a passive process. Again, no ATP is required. Diffusion is the movement of solutes from high to low concentration. So the solutes will move down the concentration gradient. And you will move until equilibrium is reached. And it is rapid over short distances, much slower over long distances, and it's directly related to temperature, meaning as your temperature increases, the diffusion rate increases. It's inversely related to molecular size. So the larger your molecule, the slower the rate of diffusion. And I've been talking about diffusion happening across a cell membrane, but it can happen in an open system as well. So no membrane is needed. So for something to move across the membrane, because our membrane is a phospholipid bilayer, lipophilic molecules tend to dissolve easily and move easily across the cell membrane. So they diffuse much more easily than lipophobic. So if you are lipophobic, you don't like lipids, you probably have to diffuse through a protein. If you are lipophilic and small enough, then you can probably just do simple diffusion across the membrane. Now, a law that's important for diffusion is Fick's law. So Fick's law states or looks at all of the properties that influence the rate of diffusion. And you have this equation here. So it tells us that the rate of diffusion is directly proportional to everything above the division line. So it's directly proportional to the surface area. So the larger the area you're going to diffuse across, obviously the faster the rate of diffusion. It's also directly related to concentration gradient. So the greater the difference in concentration, the faster the rate of diffusion. And then it's going to be directly related to membrane permeability. And membrane permeability is your lipid solubility over your molecular size. So if your solute likes lipids, you'll diffuse much quicker. But if your molecule is really big, bigger molecules move slower, so you'll diffuse slower. And then membrane thickness. The thicker your membrane, the slower the rate of diffusion. It is inversely related. Now, in your cell membrane, you have different types of proteins. Some of them help give your cell its structure. We have some enzymes, we have some receptors, and then we have transporters. Again, transporters are things that are going to allow solutes to transport or move across the membrane. And transporters can come in the form of channels and carriers. So here's a figure. Here's a channel, here's a carrier. Notice a channel is fixed and it's filled with a water pore. Whereas a carrier kind of is open on one side at a time. So it kind of moves. So here it's open on the ICF and then it'll switch and open on the ECF. And so that's how solutes will move across the membrane. Now, just because a channel is fixed and open and has this water-filled pore, it doesn't mean we can't close that channel. And we can with a gate. So here is a gated channel. Here the gated channel is open. Here we've closed the gate. Some channels don't have a gate. They're just open and kind of leaky. Other terms I want you to be familiar with are uniport and symport and antiport. So uniport means you're going to move one thing. So in this case, only glucose is moving. In symport, you're going to move more than one thing, but they move in the same direction. In antiport, you're going to move more than one thing, but they move in opposite directions. So just terminology. Now let's go back to these gated channels. How do these work? Well, normally if you have a gate and you close it, you need a key to open it. And so the same thing applies here. Some gates require a chemical, a ligand, to bind to it to open. So we call those guys a chemically gated channel or a ligand gated channel. Some gates will open when the voltage around the gate changes to a certain number. So we call those channels voltage gated channels or electrically gated channels. And then some channels, we need something mechanical to get it to open, so something physical. So we call those mechanically gated channels. So changes in temperature or changes in the tension on the membrane will get these guys. Or actually, we'll have an example later on where another receptor physically pulls the channel open. So let's review the diffusion that we talked about earlier. So facilitated diffusion, if you recall, is where solutes move from high to low concentration. No ATP is required, but we do need a facilitator, a helper. So in this case, we have this protein. The example here is a GLUT transporter. So GLUT should make you think of glucose. So this is a glucose transporter. It allows glucose to move. Diffusion will stop once equilibrium is reached. Here's an example of primary active transport, and that would be your sodium-potassium pump or your sodium-potassium ATPase, because ATP is required. How does it work? It pumps 3 sodiums out of a cell. Here, the sodium's going out. 2 potassiums into a cell, and again, ATP is required for this to happen. Why? Because normally sodium is higher extracellularly, potassium is higher intracellularly. Sodium normally has a gradient that wants to push it in. But the sodium-potassium pump can actually pump it out. How? How can it move sodium against the gradient? Because it's using energy. Then we have our secondary active transport, our SGLT, sodium glucose linked transporter. So basically, it's going to move sodium and glucose together. ATP is required in an indirect way. So this sodium-potassium pump pumps sodium out of a cell, creating this high sodium extracellularly. We're going to use the sodium that's high extracellularly that really wants to go into the cell. We're going to use that gradient to force glucose to go into the cell, even though glucose is already high inside. So essentially, sodium is kind of grabbing glucose and forcing it to go in. And this is your SGLT. And then our vesicular transport, we talked about phagocytosis, endocytosis. We also talked about exocytosis. So here is an example of a ligand binding to a receptor. It will migrate to a clathrin-coated pit that will trigger endocytosis happening. And then here we see exocytosis going on. Transcytosis is another way where we can move solutes, and we have transport going on. So in transcytosis, we will have endocytosis happening in one side of the membrane. It will then, the vesicle that endocytosed will go to the other side of the membrane and exocytose the contents to the next space. Okay, so it's where you have endocytosis and exocytosis participating together. So that's transcytosis. Transepithelial transport. This is an example that you saw in week 1, where we're going to transport solutes from one side to the other, but we're a lot more selective. We talked about this when we were talking about different types of epithelial cells. So in the intestines and the kidney, we want to bring glucose from the lumen into our body. We will use the SGLT to do that. We'll need the help of the sodium-potassium pump and GLUT transporters. But again, we're going through the cell. So that's transepithelial transport. We're not going through vesicles. We're not going in between the cells. We're transporting through the cells through proteins. Now let's move on to osmosis. Osmosis is the movement of water, and water will always follow solutes. So if you remember water follows solutes, then you know where water goes. But this only happens when the solutes are unable to diffuse. So if you have this U tube, right, this tube that's shaped like a U, inside A you have some solutes, inside B it looks like you have double the solutes, so there's a higher concentration of solutes inside B. So these solutes actually want to diffuse into side A, but it can't because they're too big. So when solutes cannot diffuse and you do not have equilibrium, then water will move instead. And so in this case, water will move into B. Why into B? Because B had more solutes, and water follows solutes. So we can look at osmolarity. So in this example, if we can say side A and side B. I would say side A is hypoosmotic to side B. So there's less solutes. We could say side B is hyperosmotic to side A. So it's this comparison. And here, they're isoosmotic. They are the same. Okay, so water will go from hypoosmotic to hyper. So in this example, water will want to go into B, from hypoosmotic into hyper. Tonicity describes the volume change of a cell placed in a solution. So if I were to place a cell into a solution and the cell swells up, why is it doing that? Well, water follows solutes. So why would water go into a cell and cause a cell to become bigger? Because there were more solutes in the cell and less solutes in the solution. So we would say the solution was hypotonic, hypo meaning less. If I put a cell into a solution and the cell does not change size, it's because the solution was isotonic to the cell. They had the same tonicity, so water didn't have to go anywhere. If I were to put a cell into a solution and the cell shrinks, it shrivels up. What's happening? The cell is losing water to the solution. Why is that happening? Because water follows solutes. So that means my solution had more solutes. So the solution was hyper meaning more, hypertonic. Okay, let's talk about electricity, and then we're going to wrap it up. So, very quickly, law of conservation of electrical charges says that the net amount of electrical charge produced in any process is zero, because for every cation, there will always be an anion. So think back to when we talked about ions. So whenever you create a cation, you also create an anion. So overall, we have a net electrical charge of zero. Remember that opposite charges attract and like charges repel. And conductors allow ions to move easily through it versus insulators do not. So in our body, the phospholipid bilayer is actually a very good insulator. Another thing I want you to know about electricity is that the inside of a cell is typically more negative than the outside. How negative? The resting membrane potential of a cell, the cells that we're going to learn in class, is negative 70 millivolts. So negative 70 inside, 0 outside. It's mostly negative 70 because of potassium, which basically tells you that when potassium levels change, your resting membrane potentials are going to change. So potassium plays a really big role here. So here's my resting membrane potential at negative 70. When your membrane potential becomes more positive, we always call it depolarizing. Okay, so whenever you become more positive, whether you're going from negative 100 to negative 70 or negative 70 to 0, if you're going in the up direction, you are depolarizing. When you become more negative, but going toward resting, you are not going past resting, you're going toward negative 70, then you are repolarizing. But if you become more negative past resting, then you become hyperpolarized. Okay? So you are depolarizing, repolarizing, you're back at resting, and then you're hyperpolarizing. All right, let's put it all together. Okay, so right now we have all of these concepts about diffusion and osmosis and electricity and polarizations. Well, what does it mean in our body? Why do we need to know all this stuff? Well, we're going to use insulin secretion as an example. So the beta cells of your pancreas releases insulin. We'll learn about that later on. But let's take a look at what's really going on. So here we have a situation where the blood glucose level is pretty low. So low glucose. Now insulin, if you don't know, causes your blood glucose to go down. It reduces blood glucose. So if our blood glucose level is already low, do we want to release insulin to lower it even more? No, because then we're going to go out of homeostasis. So we don't want to release insulin in this instance. So this beta cell, we don't want it to release insulin. So here's insulin in these vesicles. We do not want them to exocytose. So what happens is when glucose is low in the blood, here, take a look. There's a GLUT transporter, facilitated diffusion. Remember, facilitated diffusion causes solutes to move from high to low. Well, glucose is low out here, so it really isn't going to move into your cell. Now, glucose, recall, is used to make ATP. If glucose doesn't really come into the cell, we're not going to make a lot of ATP. Now, here we have a potassium channel. It's a gated channel, and some of these potassium channels that are gated are always kind of open. We call these leaky channels. And potassium leaks out a little bit, and that's okay. That's actually normal. Okay, and that keeps us at negative 70 inside the cell. So this is a normal thing. So when ATP is low inside the cell, this potassium leak channel stays open. So the leaking that's supposed to happen can continue to happen, and you stay at negative 70 millivolts. Now, this channel here is a voltage-gated calcium channel. Remember, voltage-gated channels open when the voltage around the channel changes. But because the potassium is leaking out and we stay at negative 70, the voltage does not change, so this voltage-gated calcium channel stays closed. And so calcium does not move in. And calcium, we haven't talked about yet, but calcium is higher extracellularly compared to intracellularly. So calcium does not move in, and calcium, when it comes into the cell, what it does is it causes these vesicles to exocytose. It's like the signal telling it to release. But so because calcium did not come in, the vesicles will not exocytose, and hence no insulin is secreted. Now let's say you had this huge meal and your blood glucose levels started to go up. Now your blood glucose is high. So the GLUT transporter will move the glucose into your beta cell from high to low, facilitated diffusion. We're going to use glucose to make ATP. So we have our aerobic metabolism. There's our glycolysis, our citric acid cycle, the electron transport chain, etc. And so we made a bunch of ATP, 32 from every one glucose, if you recall. Now what that's going to do is it's going to cause this potassium channel to close. Now remember, when this channel is open, potassium leaks out, and that allows us to stay at resting, -70 inside the cell. Now, potassium is a cation. It's positively charged. If we close this channel, those positively charged potassium ions do not leak out, which means those pluses start to build inside your cell. So that's going to change your resting membrane potential. It's going to go from -70 and it's going to start to creep up. It's going to start to depolarize. And now that we've changed our resting membrane potential, this voltage-gated channel may be triggered to open. So if the voltage changes enough, the calcium channel, which is voltage-gated, will swing open and calcium will flow in because it will want to go from high to low. It's high outside, low inside, so calcium rushes in. And calcium will then signal these vesicles filled with insulin to exocytose, and so insulin is released and it can do its thing to cause your body to lower blood glucose. Where did the insulin come from? Well, we made it. We did our protein synthesis, so DNA, we transcribed it into mRNA, then we translated into protein, and then from the rough VR, we sent it to the Golgi, and the Golgi packaged it into the little vesicles, and they were just sitting here waiting for the right signal. Okay, so that is a summary of Chapter 5. Thank you.